ladies and gentlemen. We know that pleasure, happiness, laughter, pain, fear, and sorrow arise only from the brain. It is with the brain that we think, comprehend, see, and hear. This is a passage from the writings of one of Socrates' pupils. His statements about the functions of the brain were less based on knowledge than on philosophical speculations. For centuries, scientists have since then endeavored to unravel the mysteries of the brain, but it has not given up its secrets easily. By awarding this year's Nobel Prize in Physiology or Medicine, the Nobel Assembly of the Karolinska Institute wants to recognize the great advances in the understanding of the brain that science owes to them. The title of Dr. Wiesel's presentation is Mechanisms Governing the Operation and Development of the Mammalian Visual Cortex. I suppose it will be an unbiased account.
Thank you, David. It's very nice for me to be back to my home university, and even if this building did not exist at the time when I was a medical student here, I remember myself sitting as you students in the back listening to Nobel lecturers before me. And primarily, I often didn't understand what they were talking about, but it was interesting to see what they looked like. I have no regret leaving, as referring back to what David Ottoson said before, in part because I had the great pleasure of first having a colleague as Steve and friend as Steve Kufler, and secondly having the pleasure of working with David as over so many years. Now, one of the interests that we had rather early on was that when we had gotten a rough idea about what cells responded, the response properties of cells were in the primary visual cortex, that was in the cat, we thought that it would be interesting to see if we could learn something about the development and also what the role of visual experience was in this process of development. Now, at the time, and actually way back, it has been a long discussion, as you all know, among philosophers and others, psychologists, about the role of nature and nurture in the development of the brain. And there was particularly, way back from Descartes and Kant had the point of view that nature was very determinant, was the determinant factor, whereas Berkeley and Holmes and many others felt that the empiricists, so to say, felt that experience was crucial. And some of the discussion that behind these philosophical arguments were based on observation in children born with cataract, because it was known quite way back that a child operated with a congenital cataract operated on, even if properly refracted, had poor vision. And the child never seemed to really regain normal vision in the eye. And of course, depending on the point of view, you could interpret that any way you like. And there was, at the time, in the early 60s, when David and I started to think about this, there were also animal experiments. Donald Hebb in Montreal had been very interested in this question and done some experiment, and a student of his, Reason, had done some monkey experiments showing that animals deprived of vision early in life did indeed have visual defects. Now, we were in an advantageous position because with the development of microelectrode, we could actually probe various levels of the visual pathway and see what the situation was. We could look at young animals and see if indeed the visual system, if the cells in the visual pathway did respond in a normal fashion. Now, we soon found that actually most of the machinery that David had described to you is present in very young animals without any visual experience. Cells are binocular, they are orientation selective, and you have a columnar kind of arrangement. And there are some differences which I will refer to later. But overall, the machinery seemed to be determined by innate mechanisms. Now then, the question we asked was, why is it that visual deprivation has such severe behavioral results? And the experimental setup we decided upon was to close by lid suture, cover the eye by lid suture. And we thought about this for a while and that we could try to raise animal in the dark, but being somewhat lazy and thinking about all the difficulties with those kind of procedures, we decided to cop out and just close the lids. And being medically trained, it was not such a big undertaking for us. And to be nice to the cat, we only closed one eye instead of both eyes. We also thought that having one eye closed, we could use the other as a normal control. So in a lot of the experiments that we started out doing, and I will discuss here, we used condition of monocular eye closure. And that's, in hindsight, that was an interesting thing, because it turns out that there is a great difference in the results, as you will see, if you close one eye or both eyes. And my guess is if we had started out by closing both eyes, we probably had said the results are so messy, let's forget about it. As it turned out, the results were quite interesting in the monocular closures. And the simplest, perhaps, way is to show first a slide of a monkey with one eye closed, just to give you a feeling for what they look like. But you cut the margins of the lids, stitch them together, and you get a very smooth surface. And this cover actually makes it impossible for the monkey to have any real form vision, but he's obviously not light deprived. And we didn't really want to light deprive the monkey, because we knew from our cortical work that cortical cells are not interested in changes in light illumination, that they were primarily interested in form, and we wanted to form deprive our animals. So what we did was to then suture the lids in the monkey at birth or shortly after birth, and then let the animal grow up. If you then, let's say after a year, examine the monkey by separating the lids under anesthesia, and then test the animal's vision in the eye, the animal turned out to be blind. And even behaviorally, that is, it would, if you cover the good eye, it would stumble and fall off a table and so on. Direct and indirect light reflexes are normal. If you record from cells in the retina, they also respond normally to light stimulation. And also in the lateral geniculate, the relay nucleus between the retina and the cortex, the cells also seem to be normal. It was only when we came to the record from the primary visual cortex that it was very difficult to find any cells responding to the eye that had been covered. So apparently, this simple experiment, by just cover one eye, has led to a very dramatic change in the cortical innervation. So even if at birth, as I said, there were binocular cells, after lid closure, all the cells seem to only respond to the normal eye, or non-deprived eye. Now, in order to assess the effect of visual deprivation, because, as you will see, it depends a little bit on the age when you deprive the animal and duration and so on, it's nice to have some gauge on by which you can measure the effects. And that's why we created something we call ocular dominance preference, which David referred to briefly. And we have subdivided cells according to their ocular preference in 7 groups. And in the next slide on the left, is shown the ocular dominance groups and in the normal monkey. Here you have group 1 cells are monocular, responding only to the contralateral eye, and group 7 only to the ipsilateral eye. Group 2 and 3 prefer the contralateral eye. That is, if you record from the right hemisphere, they prefer the left eye. Group 5 and 6 prefer the ipsilateral eye. And group 4, we really couldn't tell which eye was the best. And even if this histogram is not, and here we have the number of cells. So there are some 600 or 700 cells from monkeys, and give a reasonably normal distribution how cells outside layer 4C, where you only have monocular cells, distribute themselves in terms of relative binocular dominance. And the effect then, if you close an eye, which is shown here to the right, at 2 weeks in this case, and we kept one eye closed for a period of 18 months. Then we recorded from the striate cortex, and we found that practically all of the cells responded to the normal eye, which is the ipsilateral eye in this case. There were a few cells responding to both eyes and a few only to the deprived eye. But it was a very dramatic difference from the situation in the normal. And if we had recorded from this monkey at 2 weeks, before we close the eye, we would have an ocular dominance picture very much like here. Now the question now is it because all the cells in this category have died and only a few monocular cells survived, which is one interpretation of this result. And this is not very likely, and this is not our interpretation. Our interpretation is that, and you will see some support for that, is that the cells in these groups with input from the non-deprived eye had changed their allegiance and now are driven only by the normal non-deprived eye. The deprived eye has lost its ability to affect the eyes. Now, this very simple experiments have led then to dramatic physiological changes, but also, and the basis for these changes, we can see anatomically. And this is if we look at the geniculate afferent innervation in layer 4 C. Now, as David illustrated, you can show by an intraocular injection of radioactive material, proline, and allow time for transport that you can visualize the genicular terminals in layer 4C. And just to repeat, the normal situation is shown in the next slide. Here you see the same illustrations as we saw before, with here are then genicular afferents from the injected eye, non-injected eye, and these are parallel bands of about equal size, 0.4 to 0.5 micrometers or millimeters. Now, if we in this monkey that we just deprived had one eye closed for up to 18 months, if we inject the normal eye, then and see the geniculate innervation from genicular terminus with input for that eye, you see a picture that is very different in the next slide. Here you can see now, instead of equal size bands, you see that one set of bands, which are the one genicular terminus with input from the non-deprived eye, has expanded greatly, and they are separated by a very narrow band, which is the genicular terminus with input from the deprived eye. So the explanation then for the change in the cortical physiology presumably is based on the fact that the monocular closure in this case have led to dramatic change in reinnervation. Now this change in the innervation actually is reflected back into the lateral geniculate body in a very dramatic fashion, and this is shown in the next slide. This is a picture of the lateral geniculate body on the monkey. And this, like in human, is a 6-layer structure, and the eyes innervate alternating layer. This is a normal, deprived layer, normal, deprived, deprived, and normal. layer. And as you can see that all the deprived layers, the cells are smaller. They actually reduce by a factor of 25 to 30%, and they tend to stain slightly less intensely with crystal violet than the cells in the normal layers. This difference in size is clearly a pathological change in the sense that they are not of normal size. At the same time, when you record from these cells, they show the typical on or off center type receptive field. So it's not reflected in their physiological response. So at least at present understanding, there is the physiology of these cells is reasonably normal. The reason for the reduced cell size had been suggested by Ray Guillory originally, that it may be a reflection of the fact that the terminal arborization of the geniculate cells from the deprived layers is smaller than normal. And the diagram in the next slide sort of is just a way of summarizing what I've just said. This is distribution of, this is a normal cortex where you have a right eye column, left eye column, and so on. And the geniculate terminals then innervate here in layer 4C. And the cells above and below are dominated by the right eye, left eye. In the deprived situation, you have, this is a change in distribution. There is the size of the two left and right eye column is the same as here, but instead one has expanded on the expense of the other. So geniculate terminals from the normal eye come in and branch and innervate this part, whereas those from the deprived layer a much smaller part. And of course this is also reflected in the upper and lower layers, which receive input from the cells in layer 4C. So therefore, when you make a penetration through the cortex, it's not surprising to find that the large majority of cells respond only to stimulation of the normal eye. It is an interesting thing to remember that the cells sitting up here, they don't really care if a cell here receives input from the right eye or the left eye. They just take whatever they got. Are perfectly happy. So there is no striking abnormality in the cells in these layers, except for the fact that they have lost their normal ocular affinities. And it's only in the region just above the deprived patch of 4C with input from the deprived eye that you sometimes find abnormal cells giving weak responses or slightly abnormal responses. So this then was the original observation we made first in the cat and later in the monkey, that by a very simple procedure you could dramatically rewire the visual cortex without making lesions, just by change the animal's visual experience. Then, of course, having found that and having done this in the young animal, we wondered, is this a property that is present in all animals? Is it an age-dependent, and so on? And by doing experiments of closing an eye at different ages in the adult and in monkeys at different ages, we could show that in the monkey there is a period of high susceptibility during the first 6 to 8 weeks, and a period with lower but still some sensitivity left up to a year and a half, and after that up into the adult. The monkey seemed to be totally resistant to the effect of monocular deprivation. So you can keep an eye closed in the adult for several years, then open the eye, record from the visual cortex, and look at the autoradiography and the geniculate innervation, and everything is perfectly normal. So the adult is resistant, and then as you get younger and younger, the animals become more and more susceptible. The interesting to think about the critical period, and if you allow me to use this term defined in this sense, is that in the cat it seems to be during the first 3 months when the animal is susceptible. In man, we know less, but certainly from clinical experience, it seems that is as if a child is extremely susceptible to cataract deprivation during the first year, even the first few months. And it seems as if a child is susceptible to various kinds of deprivation during the first 4, 6, perhaps 8 years. But again, the younger the child, the more susceptible. So that seems to be a general principle. Now one can show this in the monkey, that just a few days of closure during the first 6 weeks is sufficient to produce a marked change in the cortical innervation. And the next slide is just an example of a monkey in which one eye was closed for a period of, I think the monkey was 8 years old, 8 days old, sorry, when the eye was covered. And then the experiment was done at 20 days of age. And as you can see, this then was a deprived eye in this case. This is a normal or non-deprived eye. And as you can see, practically all the cells responded only to the normal or non-deprived eye. Now, if you take this animal now, and after the recording, revive the animal and let it survive, and see, is this change permanent, or is it possible for the animal to recover the proper cortical connections? You find that there is a very limited recovery, and the next slide is an illustration of that. This is a monkey in which one eye was closed for 9 days. It was between day 11 after birth to day 20. And at that, then at 20 days of age, the other eye, the eye was open, and both eyes were left open for a period of 4 years. During this period, the animal was tested behaviorally to see if it would regain vision in that eye. And we found that at first the animal seemed nearly blind on that eye, but with time and testing it did regain vision, so that it had maybe a reduction of normal vision by a factor of 3 or 4. It never regained normal vision compared to the normal eye, which had 20/20 vision, had more like 20/80, 20/80 vision. But there was some. But you can also see here that in the cortical physiology, that there is still, this is a normal eye, the eye was never closed, is very strongly dominant. And most cells, even if there is some slight recovery perhaps, most of the cells still, after all these years and after having both eyes open, still were primarily driven by the normal eye. So it seems as if even a closure of an eye for a very short time early in life can produce permanent defects. And I think there are some studies, one in Japan by Dr. Awaya, and some other studies suggest that indeed in children with an eye patch, because of, in Awaya's study, he did surgery of lids. The eye was covered for about a week or 10 days. And then when he read about these type of experiments, some 10 years later, he went through material and called back patients who now were in their teens or grown up. And he found that in this patient that indeed an eye that had been patched early in life, there was a decrease in the normal visual acuity. And the younger the child, the more severe was the effect. Now, there is one way by which the ophthalmologists try to improve vision in a weak eye. And this is particularly common in children with Strabismus or cross-eyed children, where you have one eye perhaps is weak, and if you cover the good eye, it's well known that you can improve vision in the weak eye. You often may then lose vision in the eye that you cover, so you have to be very careful in balancing this effect, so you don't get two eyes which are defective. And there is actually no ideal way of dealing with the situation. But in an animal experiment like this, we thought it would be interesting to see what happened if you, as we open this eye, we had closed the other eye and see if that would have made any difference. And these are experiments that also Colin Blakemore and his group has done with similar results. Now, this type of reverse suture have very dramatic effects if they are done during the early period of high susceptibility, that is in the monkey between the first 6 to 8 weeks. And I'd like us to show an example of that in the next slide. This is a monkey in which one eye was closed during the first 3 weeks, and then you had this result. And then if you reverse your eye, so the initially closed eye, which would be this one here, is opened, and this eye is closed, the D2, the secondly closed eye. And then the animal at 3 weeks of age, and then you record from the animal 6 months later, you find that it has been a complete reversal of the effect, so that the eye that could not drive any cells now drive practically all cells, and vice versa. So there we have like it's a tremendous plasticity here, that connections can be lost and collections can be formed, and depending how the visually information is manipulated. And this actually can be, is also reflected in the innervation of genicular terminals by autoradiography. Of course, you can't do autoradiography in the same animals in 2 different stages. But by having done autoradiography in 3 weeks animals, we know that at this stage there is already a shrinkage of the deprived column and expansion of the normal. And now if you look at this case, of course you can look at the autoradiography, and I like to show a picture in which this eye, the initially closed eye, was deprived. And the picture is in the next slide. Here then is the, you can see then the broad expanse of the geniculate afferent from the eye that was closed initially. This is an eye where at the time of eye opening there were narrow columns. So the fact that most cells indeed did respond to the initially closed eye is well reflected in the autoradiography. Now there is one thing here which is, came as a surprise to us, and it's a little bit detailed in some sense, but it's important from another point of view, and that is that you can see that one part here of the picture there are very narrow stripes. I think it's another area up here. Now it turns out that the cells, as I mentioned to you, there are 6 layers in the geniculate. The 4 most dorsal ones are called the parvocellular layer, and they innervate the lower part of layer 4C, called 4C beta. Whereas the bigger cells in the most ventral layer, the magnocellular layers, innervate the upper part of layer 4C, the 4C alpha. And it turns out that its only expansion is only seen in the 4C beta, in the lower part, in the parvocellular innervated part. Whereas in the part innervated by the magnocellular layer, there is, the situation is the same as at the time when the reverse suture was done. So it seems as if the magnocellular layer have lost, they had certain plasticity at birth because they did shrink. But they, at 3 weeks, when we did the eye reversal, their plasticity period was over, and they could not change affinity again. Actually, you can make a reconstruction of the innervation in layer 4C alpha and beta, which is shown in the next slide, and see how different it really is in the same area. This is then the parvocellular innervation in 4C beta, and this is the narrow column seen in 4C alpha. This is interesting in terms of the age of plasticity. Again, come to the point that neural plasticity may not be a general phenomenon in the brain, so that all parts of the brain is plastic or have ability to plasticity at a given time. But that different parts of the brain, that even within a given nucleus, cells differ, may be depending on their birth date or other things that we don't understand, they have this ability of plasticity. And it may be that other part of the brain has an entirely different kind of critical period that has to be worked out for each part of the brain and each subpart of any given structure. Now, if you do the reverse type of experiment, when the animal is 1 year of age, there is no recovery. The columns stay shrunken, and the initially closed eye is still incompetent to drive cortical cells. So this ability to recover through the reverse suture is an age-limited one and is restricted to the early part of life of the monkey. The next question that I like to come to is somewhat about the mechanism. One would perhaps think that on basis of this experiment that the animal is born with certain connections. One eye is closed and the connections are lost. Now, if we had then closed both eyes, then maybe all connections would be lost. So we would have a cortex that was more or less completely unresponsive to light stimulation because of the disuse. Now, it turns out not to be the case, because if you raise a monkey with both lids closed during the critical period, you find that you have a cortex that is indeed very active, and if you have the two eyes closed, let's say for the first 6 weeks, when we do have very dramatic effect of monocular closure, the cortex is very active and practically normal in its response properties. You have oriented cells, and they are very columnar organizations, and all the things. And the only things that seem to have changed is that you have a loss of binocular cells. I mean, this seems to be something that is very delicate in the visual pathway, is the precise balance and maintenance of the binocular connections. And I think the next slide is an example of a monkey in which both eyes was closed during the first 30 days. And as you can see, instead of the normal distributions with a fair amount, maybe as David said, half the cells being binocular, you have most of the cells either responding to the right or to the left eye. So the mechanism for this is not clear, but one thing is obvious, that the two eyes have not been used together in a normal fashion, and we believe that that is essentially an essential part for the survival of connections. But one conclusion that struck us very early on when we did this in the cat experiment was that it seemed as if if a connection is maintained from one eye, it depends, and if one eye is closed, if connections will survive, depends if the other eye is open or closed. That shows that there is an interdependence between the input from the two eyes in the cortex. And this led to us to say, forcibly, that when you talk about deprivation effects, it's not disuse really that is perhaps the most crucial factor, but this idea about competition between input from different sources. In this case, input between the two eyes. Because in other systems and so on, it may be other competing influences that will determine the outcome if you make the system, take the system out of balance. So in the monocular closure situation, you obviously create a very disfavorable situation for the deprived eye relative to the normal eye. And that apparently changed then the balance and make one set of afferents being favored over a different set. The other way by which we have manipulated the input from the two eyes has been to try to produce and raise monkeys with strabismus. And we have produced the artificial strabismus by sectioning an eye muscle, either the lateral rectus or the medial rectus. It turns out that if you serve the lateral rectus in the muscle, the eye turns in, and the muscle usually inserts itself again in the lower part a little bit further back on the globe, so you get normal eye movement, but the eyes may be 10 or 15 degrees out of alignment. Now there you have a situation where the animal in some sense is not visually deprived because the input from the two eyes is the same as in a normal case. But the input from the two eyes is no longer synchronous because the images on the two eyes, the receptive fields will no longer be on corresponding point, but you may be excited from one eye and inhibited from the other. And this lack of synchrony from the two eyes is apparently what causes a disastrous effect on the binocular connection. And that is illustrated in the next slide, which is a monkey that is 3 weeks of age, a normal monkey. We cut the lateral rectus of one eye, and the monkey had a moderate strabismus. And then we recorded when he was 3 years old. And before we recorded from this animal, we tested the visual acuity in both eyes and found that the monkey had good vision in both eyes. But we also set up a test situation so we could tell if he could fuse. That is, as you know, in kids, you ask the kid to put the lion in the cage. And these monkeys could not put the lion in the cage. So we concluded that they were alternator there, with good acuity in both eyes, but lacking the ability to fuse. Of course, you're not surprised that these monkeys, when you then record from the visual cortex, have problems with fusing, because they have no binocular cells, which we believe is, of course, essential machinery in the visual pathways for get the two eyes together and to work together. Now, so this then would be a situation that would perhaps provide the physiological basis for a situation of an alternating strabismus. As you know, there is also common in children with the strabismus that they have low vision in one eye, so-called strabismic amblyopia. And the physiological mechanism of that is less clear. We have, in our series, we have found very few monkeys. Most of them have turned out to be alternator. So I think the Van Orden in Houston and Ikeda in London and one of our monkeys showed effects as if one eye had been closed. That is, there were in the eye that was good, had more cells driven by that eye than in the amblyopic eye. So whether that is a situation that there is some sort of selective loss of ability of the amblyopic eye for some reason to drive, we don't at present time have a good understanding of. But the main lesson for us as physiologists interested in trying to think about the mechanism, how the effect of deprivation is created, is the fact that the competition between the input. Now, when you look at the physiology of these animals, you find that the cells preferring or responding, let's say, to the left eye only, they are grouped, they are not monocular then only in layer 4C. As you know, you had these patches of monocular region. But actually, the monocular region extends all across the cortex. So you have now a column of monocular cell extending from white matter all the way to the cortical surface. And it seems, our conclusion from that is that it seems as it is a dominant eye that takes over. So in the strabismus situation, you have a number of cells preferring one eye over the other, and it's always the eye of preference that seem to take over and determine if a cell is going to be driven or not. And the reason for that perhaps is that it's always a dominant eye that is going to tell the cell to fire. The non-dominant eye can say whatever it will, but the dominant eye is going to determine if a cell is inhibited or excited. And it may be important for a synapse to survive that it has some effect on the postsynaptic cell, that it fires the cell or so. In the normal situation, the weak, non-dominant input will always have the same effect as the dominant, and therefore is tolerated by the cell because the cell will think it has an effect. Whereas once you have the system out of synchrony, the weakness of that input is unmasked, so to say. The next thing I'd like to address shortly is about the development of particularly the ocular dominance columns. Now, as I said before, at birth, and when we record from the cortex, there are binocular cells. And when you do an ocular dominance histogram, the difference between the normal and the normal baby and the normal adult is not very great. Now, we were somewhat surprised when we started to study the development of the ocular dominance columns to find by our radiographic studies first that at birth or shortly after birth, in our case about 8 days, that the columns indeed were not fully formed. Actually, they were not fully crystallized out in the shape that we have seen here in the normal case until the animal was about 6 weeks old. And also Pascal Kjell, during the same period, showed that when he looked at the fetal monkey, that initially, maybe a month or so before birth, when he injected an eye in an embryo, that there was no sign of any columns in the visual cortex. So there was a uniform innervation of the geniculate terminals from the two eyes. And it was not just until just before birth that he started to see some tendency to segregation. So there is then apparently a period during which these segregation occur after birth. And I think the next slide is just an autographic illustration of, maybe we can skip this, of the, so this is a picture in an 8-day-old baby monkey in which one eye was injected at birth. And you can see perhaps, and this is layer 4C where we have the genicular terminal, and you can see a tendency to a segregation of terminals. But it's not more than a hint. And if you do physiology at this age, you find that indeed there are narrow regions with one eye only activity, but there are also large regions where we could drive cells from both the left and the right eye, something we never see in the adult. And at 3 weeks, there is a reasonably good segregation, and at 6 weeks, it's certainly complete. And Sokolov have done experiment using 2-deoxyglucose, also finds that at birth the columns are not fully formed, but they develop during the first few weeks. Now, when you think about what happens then when we deprive an eye, it's clear that if an eye is deprived very early on, before the columns have segregated, that one can think about that nerve fibers already present in the region just stay, instead of having the normal segregation, which, by the way, occurs if you keep an animal in the dark for the first 6 weeks and then look out in the autoradiography, you find that the columns have formed in the normal fashion. So we don't believe the visual experience as such is crucial for the segregation of the terminals. But if you deprive a monkey at this stage, you will find that you could say that the terminals are not segregated, and therefore they stay instead of having their usual way of segregating out. So then when we think about the effect of visual deprivation, it's not that you have sprouting or anything like that. It's just that you have a maintenance of a very primitive type of innervation. Now, the interesting thing is that if you close an eye at this stage, when we do have this segregation, you still get a picture as dramatic as when you close an eye at an earlier stage. You get the expansion and contraction. So we must conclude as well that there is this ability for these fibers in one area here, when you close the input to this, to migrate, send fibers sprouting perhaps out and innervate cell, which otherwise would be innervated by the other eye. What the signal is that give rise to this sprouting, we have no idea, but it's certainly one of the most interesting questions. Now, one way to illustrate the effect by which the columns are formed can be done by looking at single fiber. Simon Levay, with whom we did many of these monkey experiments, he also have looked in the cat on the development of ocular dominance column and shown that that also occurs over a period over several months. And the next slide is a picture from a paper by him and I think Mike Stryker. And at 15 days, as you can see here, layer 4, there's a uniform innervation. At 3 weeks, there is some tendency to segregation of terminals, but not until at 3 months or so, there is a complete, more or less complete segregation of terminals. And he has also done silver impregnation of fibers around this time when there is uniform innervation. And the next slide shows an illustration of that. In the top slide here, this is a 15-day-old geniculate afferent impregnated through Golgi method. And that expands uniformly over a very large area, including several ocular dominance columns. Now, so this is in the early stage before columns are seen. Now, this picture is from an experiment that Charles Gilbert and I have done in an adult cat, in which we injected a single afferent geniculate fiber with horseradish peroxidase, HRP, which is an enzyme that spreads very well in cells and stain all the processes. And when you react it properly, it forms a black stain. In this case, we injected the axon here somewhere, and as you can see, there is one, and all this is then one single axon. And as you see, the one cluster here, and then there are 3 fibers running across this gap, and there's another cluster of terminals here. And we believe this has to do with the ocular dominance column, one here, and this is intermediate column. So when you look then at the situation in the young and the situation in the adult, it looks as if this type of innervation is formed through pruning of afferent, as if selective deaths of terminals or migration of terminals or whatever the process is, so you end up with this pattern. Now, in the lower slide is another intracellular injection of an axon from a cat which had one eye deprived from birth. And this particular geniculate cells were driven from the normal eye. This is actually one axon. It's just that it branched below here. And in order to get it onto one slide, you have to take my word for it that all these processes are from a single axon that was injected down here. You can see now that instead of this cluster of 2, there is a rather uniform innervation over an area that would cover these 2 columns. So it could be that perhaps if this was a situation that was a sprouting, or it could be that if it was done early on, that the pruning that normally would occur selectively has not been taking place. This then is the end of my discussion of our experiment on visually deprived animal. And I think the lesson that we have taken from it is that it is meaningful to think about the nervous system as having a very well-planned innate design that is read out and is developing obviously before birth and after birth to a large extent through such processes. But also we have clearly shown that the environment is absolutely crucial. The use of the nervous system during the early phase of development is absolutely essential for the full development of the nervous system. And this we certainly have shown for the visual system, and one would like to extrapolate and think that that is also true for other parts of our central nervous system. Now, it seems to me science is in part a matter of pleasure. I mean, it's a pleasure to work in the lab and discover things. It's also a pleasure to do it because nature is so beautiful. I mean, I think from what you have seen, what David showed in particular, that the intrinsic organization of the cortex, for example, is just beautiful. And I thought of ending my lecture showing some things that perhaps has less, in this context, a reason to, I have perhaps not a very cogent reason to show it, except that it gives you another view of the nervous system that some of you may not have had the privilege to see. Now that refers to some work that Charles Gilbert and I have done in a study trying to study and look at the intrinsic organization of the columns. If you accept the notion that a column, either an orientation column, let's say, is a functional unit, then the next question you ask is, how is that functional unit built up? How do you go from concentric fields to simple cells and then to more complex cells? And what is wiring in sense? In order, I think our feeling, being reductionists, is that it should be possible in detail, down to the molecular level, to understand exactly how this is done. So what Charles Gilbert and I have tried is to, by intercellular recording, study cells, simple cells and complex cells, and in terms of the physiology, and then label them with horseradish peroxidase, HRP, do reconstructions of the cells in 3 dimensions. And I'd like first to show a few cells in the 2-dimensional, and then show a film where you can look at them in different orientation. The reason why we think it's interesting to know their 3-dimensional structure is that they have certain constraints upon them because of the columnar organization of the cortex. They should not just perhaps sit as a symmetric blobs, but they may have an internal structure guided by the fact that they have to fit into a certain system and adapt to other cells with different properties. So if the chairman allow me to just do this, we can do that for fun, perhaps, or something. So I'll just show first a cell. If you can have the next slide, a couple of slides first. This is a cell, a pyramidal cell injected with horseradish peroxidase. And you can see the cell body and the dendrite. And then in the microscope, you can trace it out and camera lucida and get the complete drawing as shown in the next slide. This is a cortical surface here in white matter. The cell body is a 5th layer pyramidal cell, the pyramid dendrite here, basal dendrite, and an axon process, and some collateral within the cortex itself. Now the next slide shows a simple cell in layer 4C that these are the dendrites. It sends its axon up to cells in layer 3, and the layer 3 cell in the next slide. Could I have the next slide? Sits up here. It's a pyramidal cell, complex in property, and it has local collaterals and an axon out of the cortex to other cortical areas, but it also provides the main input to the cells in layer 5. And cells in layer 5, in turn, as shown in the next and last slide, here's a layer 5 cell, the pyramidal cell, has a tremendous axon that goes and makes numerous terminations in layer 6. So now in the film, we will see first the geniculate afferent, and then we will look at complex cells, 2 complex cells. So this is the same afferent that you saw, the deprived axon, and you will see it now in the cross-section. Then it will be rotated so you can see it from above. And later on you will see that with the computer you can see in these drawings all the terminals. So now you're looking at it from above. As you stood above the cortex and looked down. And you can see individual terminals here, and you can ask the computer only to show you the terminals and not the processes. And this is back to the original orientation. And these are just the endings now. And sometimes can be very useful. You can see the clustered type of endings, and sometimes you can see a pattern actually in the way which cells terminate. And then this is particularly interesting in this plane, where you can see a certain structure to banded pattern. The next cell is a complex cell in layer 6, I think, of the cortex. So here's basal dendrite, apical dendrite. It's most of layer 6 cells often send their axons up into the cortex to layer 4, and this is a process going out. And you perhaps can see that it's not fully symmetric in its shape. It's like a space ship. You can stop and then you can turn it the other way as a perpendicular. Now you're looking down from the top of the cortex down on the cell. And you can see again that it's oriented. It's interesting also with these axons running in parallel and the innervation of that. Then with EM you can take out these sections and look at the termination on which cell types. This is this amazing fifth layer pyramidal cell with the cell body here. This is cortical surface here, and the cortical surface runs something like this. This axon goes down to layer 6. This is an axon that goes into white matter away to the superior colliculus. You can see as you turn it, it goes away in a different direction from the others. You can see how asymmetric it is. See, in this plane it's practically flat. So as we learn more of these are all normal cells, but we are at present time looking at cells which have been deprived, and we want to know in which way and how the cells are being affected by the environment in which they grow up. So thank you very much.